This is part two of our chemistry series with Dr. Paul Daley. Some of you were here for part one, some of you are coming in at part two. And I would love to introduce Dr. Paul Daley, who has been managing the lab at the Shulgin Farm for 19 years. And we will have questions at the end, last 15 minutes or so, and our last part will be on November 18th. I'll send out a reminder as well at the end of the session. So, Paul, I will hand it over to you.
Okay, thank you so much, Stacy. So for anyone who's new to this series, we're covering the basics of psychedelic chemistry, but with a sprinkling of history and human effects and how did we get here? So as Stacy pointed out, I've been working here at the Shulgin Lab, and this in fact is not a fake background. I am sitting in the same spot where Sasha did a lot of his greatest work. And with that, why don't we just climb into the presentation? So I'm not sharing just yet. Let me get that straightened out. Can you see my screen?
We can.
Very good. Thank you, guys. Okay. All right. So this is the second part. In the first part of this series, we discussed phenethylamines, which are the family of compounds that are most closely related to mescaline. That also includes MDMA and 2CB and a lot of our favorites. But today we're going to talk about lysergamides. And the lysergamides are kind of a different bag in a way because the history of how humans have been affected by these compounds goes way, way back. I didn't include all of the most uh, early descriptions of. I'm just struggling a little bit to make sure that I can control this here. Here we go. Okay, so the lysergamide. Let's give a little bit of a background. The lysergamides are compounds that are derived from lysergic acid, which is a four-ring structure that's found in a group of fungi, including the genus Claviceps, which is an important parasite of grasses. Also in the seeds of some convolvulaceae, those would be morning glories and Hawaiian wood roses, and in microfungal associates of some grasses. Ergot alkaloids are today used as pharmaceuticals, which of course includes LSD, but they're all semi-synthetic. In other words, we have to start with the four-ring structure that we derive from some natural source, and then we modify that to create specific compounds that we put into pharmaceutical use. So in broad terms, the lysergamides are mimics of naturally occurring neurotransmitters that are found throughout our bodies. So serotonin and dopamine are probably the two principal ones that lysergamides interact with, to a much lesser extent with epinephrine or adrenaline. Now, there are broadly three classes of biological activity that are important in the lysergamides. The one that has caused the greatest grief and human suffering are the compounds that cause vasoconstriction. So on the left, you'll see ergotamine and
Come on,
ergovaline.
Jesus fuck, what are you doing?
Was there a question? No. Okay. So ergotamine and ergovaline are also referred to as peptide lysergamides. So see if I can use my arrows here. So the four-ring structure that's common to all of the lysergamides is this ergoline nucleus here at the bottom. But these vasoconstrictive compounds also have a peptide ring that's attached as an amide. So ergotamine was the first of the lysergamides to be isolated and identified chemically. Ergovaline is one that's found in those grasses that I mentioned earlier. And these cause the peripheral blood vessels to constrict very strongly. We'll get into what effects those have had. So in the center is another major biological action, and this is oxytocic action. This is the causing of a strong and sustained constriction of the uterus in women. Uh, this is so strong in effect that, in fact, following childbirth, the bleeding that often occurs following parturition can actually be stopped by administration of ergot preparations or ergometrine specifically. And on the far right, we're also interested in the psychedelics, LSD 25 being the most well known, but ergene right next to it is also somewhat psychedelic and is found naturally occurring in the convolvulaceae, for instance, the Hawaiian wood rose seeds and morning glory seeds. So in all of these, there's an interesting conformational issue. The active forms of these have a configuration at the top of the ring, which you'll see up here, where, if you will, this acid group sticks out towards us, comes out of the plane of the paper, if you will, and that's contrasted with the isoforms, where the conformation of that group is flipped, so it's now in the back of the plane of the paper. I've tried to show that down here in a sort of a 3D view, but it turns out that these natural D forms are where all the biological activity is. The isolysergic acid-derived lysergamides, by and large, have no activity, although it's a little bit difficult to study because they always seem to be present in one ratio or another. They're interconvertible. So, for instance, if you've made an LSD preparation, there's always going to be some amount of iso-LSD present, and that, in fact, may alter the net subjective effect of the drug. But this can be controlled, and they can be relatively easily separated. So, why are these things so active? Once the structure of LSD was known, which really was when Hofmann prepared it in 1938 and then discovered its activity in 1943, pretty quickly it was noticed that there were substructures within it that resembled other neurotransmitters and other psychoactive compounds. Now, it turns out serotonin was not really discovered until some years later, in the early 1950s. But there is a fraction of the ring structure that looks serotonin-like. If we had a hydroxy group sticking off here, this would almost be exactly serotonin. And yet, if we look at another portion of the molecule, there's also a phenethylamine-like structure. So these molecules fit in the receptors in our nervous system very, very tightly. They migrate in there and they're held sort of snug as a bug in a rug, and they stay active, firing off those receptors for a very long time. So this is a very simplified diagram just to remind. The incoming nervous signals would come as impulses down from that axon, and then they force the release of neurotransmitters, which have to diffuse across a cleft and then bind with receptors on the neuron at the bottom. So the drugs that we're interested in also bind with those same receptors, and they can either turn them on, they can antagonize their action and turn them off. But in any event, these interactions are very, very specific. It turns out that LSD's action is probably mostly at serotonin receptors, but LSD hits receptors for serotonin that are found throughout the body, not just in the brain, but in the gut, the circulatory system, and other parts of the body as well. So, since we're interested in the psychedelics, Their recent history is very important, but ergot, as the source of lysergamides, has had a very, very long history in human biology. So, as I mentioned, going back as far as the Assyrian civilization, 1,000 years or so before the current era, there are accounts of pustules in wheat that caused sickness. And by the Middle Ages, when rye was in fact the primary bread grain of the central part of Europe and very, very susceptible to ergot infection, this caused huge, huge suffering. There were literally hundreds of thousands of people that chronically consumed ergot contaminated bread. And since it wasn't known why these people were sickening and dying, there was really very little that could be done. Uh, by something like the year 300, monks in the Order of St. Anthony started opening up treatment centers, early hospitals, if you will, to treat victims of ergot poisoning. And these numbered over 300 across the center of Europe by the late 16th century. But to summarize, there were a few forms of sickness caused by chronic exposure to ergot. The most spectacular was gangrene and associated loss of limbs from the vasoconstrictive aspects of gangrene. But there was also a convulsive form and a hallucinatory form that's mostly associated with acute consumption of large amounts of ergot. In any event, this was next to the bubonic plague, probably the most serious human health problem of that whole historic period. So Saint Anthony was associated with the situation of Saint Anthony, what was called Saint Anthony's fire. And in this woodcut on the left, you'll see that there strangely were offerings to St. Anthony and some of these early hospitals of limbs that had fallen off. So if you look at the poor fellow in the lower right there, he's already missing his left leg. Well, he might have donated that as a reliquary to the hospital. In any event, the first technical descriptions of ergot date all the way back to the 16th century. There's a writing from 1582 at the lower right. So this was recognized finally, and the association with contaminated bread did start to spread and become better known, but it took a great long time. Meanwhile, there were other beneficial uses of ergot that were discovered kind of parenthetically. Midwives discovered that ergot could be used during childbirth. And at first, it was used to hasten childbirth that was proceeding slowly, and also to uh, halt bleeding that would follow after a troublesome childbirth. The collection of ergots that were known to be relatively safe was then actually a fairly organized practice starting in the mid 16th century. So that makes ergot the first microorganism that was ever cultivated for a pharmaceutical purpose. But by the mid 19th century, it was realized that the practice of using ergot to hasten birth was more hazardous than it was worth. This gentleman, David Hossack, here in the United States, first suggested that the likelihood of there being stillbirths caused by too early or too strong of dose of ergot really outweighed the benefit that could possibly be had. And so he suggested that ergot be restricted to treatment of postpartum hemorrhage. Interestingly enough, he was also the physician that was called to treat Alexander Hamilton after his fatal duel with Aaron Burr. Now, all of this happened really before the whole life cycle of ergot was understood. That took place in 1853 by Louis René Toulaine. So ergot is a parasite, And the ergots that we normally see, which you have illustrated there at the lower left, are overwintering stages of this fungus. So the life cycle starts when those overwintering stages in the early spring, after being exposed to cold and moisture, sprout these tiny little mushroom bodies that are called stroma. The surface of those are covered with little pockets called perithecia, and sexual spores called ascospores are released, and they spread to flowering cereal grains and wild grasses. They germinate. They kill the growing seed of a grain and take over. They form an infection plate, which fools the plant into pumping sap, if you will, phloem sap, containing sugars and amino acids through that plate. And the growing fungus sheds conidia, asexual vegetative spores, into that liquid, forming a sticky honeydew, which you see there in the photograph at the lower right. Now, that gets splashed on by rain or picked on by insects and spread to other plants. So this becomes a secondary infection cycle that can continue pretty much throughout the summer. As conditions cool in the fall, the fungus starts a shift in its morphology and starts to make these very compact, hard sclerotia, which eventually fall off the growing seed head, land on the ground, and wait for winter conditions to activate them for germination in the following spring. So, with that information in hand, ergot began to be cultivated commercially on a fairly regular basis. And a lot of this happened in Switzerland and other Eastern European countries, but it was a very laborious process. Often women were hired to do hand inoculations of rye plants with known races of ergot. And eventually it became mechanized, as you see there at the lower right. So with this kind of host plant dependent uh, biological production, there was still many thousands of pounds of this that could be produced each year, but it really didn't satisfy the international demand. So that had to wait before modern science kind of took over and created some new methods of cultivation. And a lot of that started in Switzerland. So Sandoz Laboratories, which got its start in life as a dye manufacturer, started to shift into production of natural products for pharmaceuticals in the late 19th century. This gentleman, Arthur Stoll, was hired by Sandoz in 1899 and started a program of looking into the components of ergot, which had been isolated but never in satisfyingly pure form. He was able to finally isolate pure ergotamine in 1918. And he made the, I think, pretty brilliant decision to hire a young natural products chemist by the name of Albert Hofmann in 1929. So they went right to work on trying to discover what principle of ergot was responsible for the oxytocic effects that were so desired in obstetrics. So around the same time, Stoll and Hoffman in Basel and John Chaucer Moore in England discovered ergometrine in water extracts of ergot. Uh, Very quickly thereafter, Hoffman, who was interested in developing semi-synthetic methods for producing any of the variety of ergot alkaloids that were known at the time, uh, also started to work on ergometrine. So that was proceeding nicely. Stahl and Hoffman started to publish patents on the methods that they were discovering, and Hoffman was set on to the problem of making quite a variety of ergot alkaloids using this new semi-synthetic technology. Let me go back one slide. So in this series, one compound that they tried to make was an analog, if you will, of a known pharmaceutical called nicotinamide, also called chloramine. So nicotinamide was a stimulant of the respiratory system. It was actually one of the early performance-enhancing drugs that athletes were using. And so the theory was that if they could make a similar amide using lysergic acid as the base, that it might be a circulatory system stimulant. And so using some of the new technologies that Hofmann had developed, he made the 25th compound in this series, which came to be known as LSD 25. Now that was made in 1938. It was sent to animal testing, and it was really unimpressive. The initial results were inconclusive, but it didn't seem to have the kinds of properties that they were looking for, and so it was shelved. Now, normally in pharmaceutical industry, if a compound doesn't show the properties that you're after, then it kind of dies. And for some reason, Hoffman, 5 years later, in 1943, decided that there was maybe something missed, something that needed to be revisited. He's described it as having had a peculiar presentiment that something needed to be further evaluated. So in April of 1943, he made a new batch, and one day he felt strange at work and decided to go home. And he relaxed, laid down, and had some interesting kind of visionary experience that lasted a couple of hours. Now, LSD effects generally last much longer than that, so it's really unclear whether he actually absorbed any of this new compound or if maybe some spontaneous mystical experience was involved. But in any event, he decided on the following Tuesday, April 19th, 1943, that he would consume a small amount of this to see if, in fact, it caused some psychic effect. He decided to take the smallest amount that could be conceived to have any effect at all, a quarter of a milligram. Now, he was a very fastidious Swiss chemist, fully aware that many of the ergot alkaloids were highly potent. Their psychological effects were unknown at that time, but certainly low milligram quantities could be, considered reasonably to be something that you could feel. Of course, now we know that we dose LSD in micrograms, and a 250 microgram dose of LSD is certainly enough to experience pretty much all of the range of effects that this compound can produce. So, of course, Stoll was informed of the outcome of this experiment, didn't believe it. The rest of the management of Sandoz also were informed. They didn't believe it. Finally, uh, Hoffman's lab assistant became the next patient to receive some of this substance, and she confirmed that, oh yes, this is highly active, and there's a lovely story of her having a trolley ride home that must have been quite spectacular. So Sandoz realized that they had discovered something quite remarkable, and they freely distributed LSD at no cost to psychiatrists worldwide to try to understand the substance better and to try to determine whether there was likelihood of reasonable medical use of this substance. So clinical reaction was highly positive. In Canada, the United States, and Europe, there were many psychiatrists who were treating obsessive patients, chronic alcoholics, people with deep anxiety syndromes, and the results pretty universally were very, very positive. In fact, Humphrey Osmond in Canada, who's partially credited with coming up with the term psychedelic, claimed that the most intransigent alcoholics that he treated, half of them 6 months later, even 5 years later, were entirely abstinent. So there was a lot of interest. There were, in fact, international meetings as early as 1955, later in 59, that drew together psychiatrists and psychologists who were using LSD in their practices. And although they still felt that they had a lot to learn about how to manipulate this substance, they agreed that it was powerful. It brought psychological theory kind of to a practical, uh, readily reproducible state, and there was great enthusiasm for moving forward with it. Of course, at that time, the intelligence and military communities also started to take great interest. The Army mounted a program for at least 11 years looking at tropane alkaloids and LSD as incapacitating agents, with the idea that if you could deliver these substances to your military opponents, that you could render them non-compos mentis, incapable of being able to carry on military activities. And pretty shortly thereafter, that was mostly in the early 1950s, news got out about this amazing substance, and the intelligentsia and many notable celebrities partook of LSD experiences, and they include some notable characters like our President John Kennedy, Cary Grant, and Claire Booth Luce, who convinced her husband to cover this in a positive way in Time magazine and Life magazine. So Hoffman, in the meantime, became quite famous, uh, perhaps to the consternation of Arthur Stoll and the rest of the Sandoz management. But others came to him with questions about natural products that produced visionary experiences. So Hoffman became acquainted with Gordon Wasson, who you see there at the microscope, in the mid 1950s, when Wasson journeyed to Mexico and became the first white person to partake of psilocybin mushrooms at the Velada of Maria Sabina. Well, Wasson also learned of the morning glory seeds that were also used in ceremonies there in Mexico, and brought those to Hoffman. And being the expert in ergot alkaloids, Hoffman was perfectly capable and rather amazed to find that these green flowering plants produce the same alkaloids that were found otherwise in only in microorganisms in the claviceps family. So they did quite a bit of exploration botanically, discovered that morning glories like Revia corymbosa, Ipomea tricolor, the seeds of which you see there at the center bottom on the toward the right, and Argyro nervosa produced rather substantial amounts of the simple water-soluble ergot alkaloids. And this was quite a surprise. Well, now we suspect that at least some of these plants are infected with microparasite endophytes, fungi that live within the cells of those host plants and are actually responsible for producing the alkaloids, which is kind of a more understandable source. But it turns out in Revia, at least, it's been shown that the production of the ergot alkaloids can take place even if the plant's been treated with fungicide to kill all endophytic fungi. So there's still some mysteries to be resolved there. So after World War II, there was a growing demand with growing populations who are coming home from the war to have better supplies, more reproducible supplies of ergometrine to assist in childbirth. By the 1930s, the rate of maternal fatality from the complications of childbirth were as high as 1 in 20. Although generally speaking, they are a little better than that, maybe 1 in 100, but still, that's an unacceptably large mortality factor in childbirth. So this gentleman, Boris Chain, who had escaped from the Nazis in Germany in the late 1930s, was hired by the Italian government to staff a program at the Istituto Superiore di Sanità in Rome to develop facilities for uh, exploration and exploitation of fungi, in particular for the development of pharmaceuticals. He was already fairly well known in that as a young, promising biochemist, he worked with the penicillium fungi that were so spectacularly successful in producing penicillin, which was really kind of a secret weapon during World War II. So he went to work developing the laboratory for microbiological production of pharmaceuticals, and their first target was ergot. So strain selection, of course, was the initial barrier and challenge. Now, the selection of strains of ergot to produce alkaloids is made a little bit easy in that the ergots can be detected and quantitated with simple optical means. So on the left, three spots of ergot-containing material. At the far left, we had spots of 10 micrograms of ergometrine. In the middle, this is a strain of, or an extract from a strain of ergot. And on the right, we have 10 micrograms of ergotamine. And we're viewing these at two different wavelengths of fluorescent light. And the ergot alkaloids themselves are highly fluorescent. And in some cases, they actually fluoresce with slightly different colors. So this gives you a great way of getting at the qualitative components of a given strain of ergot. And then on the right, we've done a colorimetric analysis of total ergot alkaloids. In this case, this is done as a standard curve with ergotamine. But this same process, reacting your extract with para-dimethylamino-benzaldehyde and strong acid, produces this lovely purple-blue color, which can be read with really quite inexpensive spectrophotometers in the laboratory. So the tools were there to screen for alkaloids, but and then it turned out that the fungus itself was relatively easy to cultivate. So at the far left, I've taken ergots and sterilized the surface, broken them in half, and put them on nutrient agar, and gradually outgrow these interesting colonies of mycelium. And as you can see from the center slide, there's quite a bit of variation that emerges immediately. But still, you've got the problem of going through all those cultures to determine which ones of them are producing alkaloids and which of those, in fact, are alkaloids that you're interested in. At the lower right, you'll see one thing that sped things along. Some of these ergots, in fact, can excrete ergot alkaloids into the medium that they're growing on. So if you look kind of closely, you'll notice that these two tubes differ in that the one on the right is blue. This photograph was taken under UV light, so we're actually seeing fluorescence of the native alkaloids that are in that produced by that strain. So this was used to mass screen for ergot production in agar slants. And then eventually this all had to be moved over into submerged culture, where basically a broth solution is inoculated with the fungus and then either ground up and extracted in the case of Claviceps purpurea, or, well, we'll get to Paspali in a moment. Uh, the problem with purpurea, it grew very nicely under submerged conditions, which is how the penicillin was being produced, but it didn't really produce any alkaloids. So where you might have on the order of 2 or 5 milligrams per liter of total alkaloid produced by Claviceps purpurea, it was really not practical to scale that up to thousands and thousands of liters in a large submerged tank fermentation. So by 1960, though, this problem was very neatly solved with the discovery that another species of ergot, Claviceps paspali, was able to manufacture grams of alkaloid per liter. And in fact, as an extra bonus, it produced the water-soluble, simpler ergot alkaloids, and the fungus mycelium actually excreted that into the medium. So recovery of it was very much eased by that property. It's a very odd little plant. At the slide in the center, you can see the honeydew that's characteristic of all of these in their vegetative state, but looks completely different from the purpurea. They're not purple. They look like little cauliflowers to me, uh, and it's a delightful little plant. Okay, so now we're going to dive a little bit into chemistry. So Hofmann's earliest LSD syntheses used anhydrous hydrazine, this NH2 compound at the far left, to hydrolyze off whatever group was attached to the lysergic acid moiety, creating a hydrazide. That's further reacted with sodium nitrite, producing the lysergic acid azide, which then can be decomposed to an isocyanate and ultimately reacted with ethylamine to produce lysergic acid diethylamide or any of the other um, desired ergot alkaloids. Well, this is practical, but organic azides tend to be a little hazardous to work with. They're highly toxic, and if treated badly, which can be as little as giving them a little shock, they may explode. Not desirable in the laboratory. So Hoffman looked into some other methods to approach the problem and found that the extra groups that you really want to remove could be easily hydrolyzed off with potassium hydroxide and gentle heating. So that gave them a pretty much unlimited supply of lysergic acid from which they could manufacture any of the other ergot alkaloids. So this became one of the kind of neater methods. This was published in 1973 by a gentleman by the name of Johnson. The lysergic acid is first reacted with phosphorus oxychloride, which is kind of an activating agent. It donates a chlorine and displaces the hydroxy group of the acid moiety of lysergic acid, which then can be very readily attacked by any amine. In this case, I've got this illustrated with diethylamine, creating, oops, lysergic acid diethylamide. Not, this is wrong. Did I do that? Yeah, I did that. In any event, uh, this shows this process in action. On the left is what the reaction mixture looks like when you've added the phosphorus oxychloride and diethylamine. You end up with a very dark colored solution that is warmed for only 7 minutes and then rapidly quenched by adding ammonia. Then there's kind of a lengthy process of cleaning that up and extracting the LSD product from all of the other contaminants and impurities. And when you get down to doing a final removal of solvent, this material has the property of puffing up into a stiff and solid foam. So it's almost like you're making a piece of pink styrofoam at that point. So that was a method that was developed in the 70s, but at that point we were already well into the middle of the war on drugs. Uh, the panic that ensued with the rapid acceptance and popularity of LSD in the 60s and 70s led the United States government and eventually many other governments around the world to ban the possession and sales of LSD, except in the hands of a very few researchers. So these two gentlemen, David Nichols at University at Purdue University and Richard Glennon at Virginia Commonwealth University were pretty much the only two scientists in the United States who had active programs looking into the mechanism of action of LSD and other psychedelics for the next 25 to 30 years. Their work was instrumental in identifying the uh, receptor systems that these drugs interact with, and their work foreshadowed both the development of antidepressants like the Prozac family, the selective serotonin reuptake inhibitors, and also new and important molecules that are directly psychedelic. So part of that work has now emerged with a very detailed understanding of how these materials dock with the receptors. In 2017, this very detailed representation of LSD nesting in the active site of the serotonin 2B receptor was worked out by Wacker and his associates, including David Nichols and Brian Roth. And we know now that when LSD hits the receptor, it changes the conformation of the whole protein, and a little lid forms that kind of traps the LSD molecule in there so that it kind of bounces against the receptor and continues to fire it for many, many hours. And this partly contributes to the prolonged action of this drug. So clinical discoveries keep happening. Sometimes they're fortuitous. Uh, it was around the year 2000, a sufferer of cluster headache, which is one of the more awful neurological disorders that people have. They're called cluster headaches because during a period where a sufferer is experiencing these, they may go for weeks where they have several attacks a day and they're incapacitating. They're very distinct from migraine. Migraine sufferers basically want to kind of hide in a dark room, but cluster headache sufferers they're very kinetic. They pace, they bang their heads in the wall, and they experience pain that is described as being worse than childbirth. About 6 times as many men as women suffer from these, and for history, there's been no real treatment. Well, kind of by accident, around year 2000, a gentleman in the UK who was a sufferer was given, well, we don't know, it was either a dose of psilocybin or LSD around the time that his cluster period would have opened, and no cluster. So he experimented personally and found that if he had even a low dose experience at the time when his cluster window would open, the whole cycle could be blocked. Well, this was kind of amazing. Uh, Two scientists, Torsten Posse and John Halpern, Posse from Germany and Halpern at Harvard, were approached by a patient advocacy group, Cluster Busters, with the idea of doing an LSD experiment to see if they could duplicate this. And they were encouraged by the Harvard medical establishment. The Harvard dean said, "Well, we had Leary here," and uh, they were encouraged to investigate other similar compounds that might have the same effect. Posse in particular thought that that was unlikely, but he suggested, well, there is this bromo LSD that Hofmann patented back in the 1950s as a possible migraine drug. Never worked out. And we think that the psychedelic factor is necessary for this blocking effect against clusters, but he decided to go ahead and do an experiment in any event. And to his surprise, found that BOL 148 completely blocked in the same way that LSD or psilocybin blocked. Uh, Unfortunately, being an old compound, they were never able to successfully uh, get this through any kind of rapid uh, approval by the FDA, and so it still remains kind of a curiosity, which is very unfortunate, but it's kind of one of the vagaries of pharmaceutical development. So new LSD variants continue to emerge. In order to better understand the nestling of the LSD molecule in the pocket of the receptor, Nichols group synthesized this azetidine derivative and actually all the variants, methyl groups sticking up, sticking down, up on one side, down the other. And they found that this configuration was the only one that preserved the high potency of the LSD effect. So very, very detailed and seemingly minor modifications of the molecule have been performed to probe how the receptors actually work. Two molecules in the middle emerged in kind of the clandestine chemistry world. Alad is apparently a bit milder than LSD and shorter acting, lacking in some descriptions some of the sinister aspects of LSD intoxication. Ethlad, there to its right, is even more potent than LSD. And even more recently, we see prodrugs like 1P-LAD, 1-propyl-LAD, uh, that are basically designed to skirt the legal proscriptions against LSD itself. So, in some cases, these drugs become useful objects of research in their own, but they address a demand in the gray market. So that's the basis of my presentation. So I'd love to answer any questions if I can. Uh, this is being sponsored by the Shulgin Foundation. We're a nonprofit. And if you're interested in any technical information, you can contact me at my shulginresearch.net email or contact us for further general information.
Thank you
So
so
with
much,
that,
Paul.
I'll stop the share. And this, in fact, is the object of our interest. This is a bottle of ergot that I grew in my garden about 15 years ago. Most people haven't seen it. It's not that hard to grow, but you do have to kind of know what you're doing. So, any questions? I'd be happy to try to answer them.
Paul, this is Stacy. Thank you so much for taking the time to do this. I've been collecting some questions and I wanted to just start with the questions that came first and then we can open it up or we'll do one in one. I'll take one for the chat and then we can open it up. Um, I wanted to start with, let's see, some of these were answered. I see as a post-grad with a degree in biology and mycology, this person wanted to work with strains of claviceps and what universities would provide such an opportunity.
Excellent question. Most of the universities that are still have programs on claviceps are doing so because they cause problems in veterinary medicine. So there are. I would have to maybe get back to that person. If you could leave your information, I can try to get a list together for you. But particularly in the East Coast, where conditions are a lot wetter, these claviceps can cause problems in poisoning of horses and cattle and whatnot. If you're interested in the human medical aspects, most of that work is being done in Europe.
Awesome. Let's open it up to the group. If someone wants to unmute themselves and ask a question, now's the time.
Yeah, I have a quick one. Thank you. That was a fantastic presentation. I was wondering if the agonism of a lot of lysergamides in intestinal serotonin receptors is responsible for the common appetite suppressant effects. Is that known?
It wouldn't surprise. That method that I mentioned that was produced by Johnson et al., the phosphorus oxychloride method, came out of a paper that was looking at the emetic properties of substituted lysergamides. So they do cause emesis, and so yeah, it's very very possible that they could be causing gastric distress. LSD doesn't seem to do that, but there's so little that's going into you, you probably wouldn't notice. Although all psychedelics have the sort of risk of causing a bit of nausea. LSD is not really known for that so much as something like mescaline, but there are serotonin receptors in the gut that, if stimulated too much, will make you feel nauseous.
We had a question from
Thank
Wed. Thank you for your question. We had a question from Wed. Is there an established synthetic route to make LSD completely synthetically?
There is, but it's really not economically practical. I think Kornfeld was the first to do a complete synthesis of LSD from raw stocks, raw chemicals, but it's about a 12-step process with great losses all the way along the line. So you start with a kilo of ingredients, you might end up with half a gram or something of finished product, which is uh, uh, impractical economically and in terms of time. So it's almost always prepared from something that's been extracted from a microorganismal source.
Awesome. It looks like we have a question from Aliciana.
Hi, Luciana.
Hi, Paul. I was just wondering if you would be able to talk a little bit about the like lysergamide migraine medication. Was that mostly for the like blood clotting effects, or was that trying to hit on another kind of receptor?
Yeah, as far as I know, okay, so she's talking about ergotamine, for instance, and then later another smaller molecule, kind of closer to LSD in size, but also a lysergamide, methysergide. Um, Both of those were kind of go-to preparations for migraine for many years. I had migraines as a teenager and was prescribed each of those. They've now been kind of supplanted by other compounds that we'll probably talk about next time, the triptans, which structurally are much closer to psilocybin, as it turns out. But the action of the lysergamides was mediated via effects on blood flow in the capillary system. So it's thought that migraine causes kind of a lack of tone, if you will, in the capillaries that surround the brain, causing this horrible headache phenomenon. Methysergide was ultimately taken off the market because uh, it stimulates the serotonin 2B receptor chronically. And if you're taking it as a prophylactic, where you need to redose several times a week, the 2B receptor starts it suitably stimulated, starts to cause proliferation of the cells where it resides, and that causes the heart valves to thicken and become ineffective, and they basically have to be replaced. Uh, the problem with ergotamine also is if you did start to take it chronically to prevent migraines, you run into the same problems that people had in the Middle Ages of having gangrene, blood flow suffering in your extremities, and you don't want your nose to fall off because you're trying to treat your headaches. It's a bad idea.
I have a question, Paul, about purity of LSD. I've heard differing concepts of if iso-LSD or too much of it is less left in the final product, that some people feel like it takes up receptor space, so the DLSD can't fully engage, and other people feel like it it's it's not close enough to that receptor to really do anything. And other people say it sort of washes out the true effects of DLSD. What's your feeling about it?
Well, a lot of this kind of boils down to feelings because those are difficult experiments to do.
Right.
This is a very old argument. My feeling is that you should only really be seeing large amounts of iso LSD if the LSD's been sort of sitting around, kept in warm conditions or in humid conditions. There is interconversion, And so iso-LSD will build up over time. Now, whether it competes in the receptor site itself, whether it has agonistic properties, whether it binds to another part of the serotonin receptor, all of that's kind of unknown. You can imagine that, well, how would you do this? Take 100 microgram as your standard dose of DLSD, and then add mix different ratios of supposedly inert iso-LSD, and then see how much effect is produced. Well, how often are you going to do those experiments? Does the person who gets the dose on this day, is he coming to the experiment on day 5 with the same psychic state that he had on day 0? These are very tough experiments. You might be able to get at it from purely in vitro point, looking at shifts in binding affinities, but very tough, and I'd say it's left unresolved, left to the student as an exercise.
Thank you.
You were saying it's hard to control the isomerization, like you don't know how much percentage you're basically going to come out with on the back end. Is that due to the starting material, or is that a process of the reaction?
It's actually a process of the reaction itself. The different routes have the propensity to produce different relative amounts of LSD and iso-LSD. But they're pretty easy to separate chromatographically. So in kind of the classic 60s and 70s era, column chromatography was used to separate them. You can definitely see them separate in thin layer chromatography. So it's a matter of were you a sufficiently diligent chemist to do the extra work to make sure that you were only releasing LSD, or did you throw everything into the pot because that stretched your grams out the door? Yeah, it's cleanliness is next to godliness kind of proposition.
No, that makes sense. Yeah, but I just know sometimes starting material has a lot to do with that. So just curious where that along the way.
Well, I've heard it said that the isoforms don't exist naturally, that they only start to accumulate once the alkaloids are extracted or they've gone through some chemical process.
Cool.
So, as you might imagine, some of these things are a little difficult to get at, but certainly analytically, you always see these things in pairs. You'll see it in the terminology is kind of fun. Like ergotamine would be the D form, and ergotamine is the isoergotamine. Ergometrine, ergometronine. So you always see these things to one degree or another.
Well, Paul, we are at the end of time here, but we had some amazing questions come through. I know some we got to, some we didn't. And I wanted to thank everybody here for joining. Paul, thank you so much for doing this class. We are going to be doing part 3, the final part, on November 18th, the Wednesday, and the event page will be up about 6 weeks in advance. And this is being recorded, so we'll post that on our site, on our YouTube page as well, in about a month. Thanks, everyone, and take care. Thanks for joining.
Thank you, Paul. Thank you
Thank
all.
you. Thank you so much.
Thanks, Paul.
Thank you.
Thank you, Dr. Paul.
Oh, thank you, Stacy.
Thanks, Paul.
Thanks so much.
Look for it.
How much is in that?
It's everywhere.